Before we get started, I have the extreme pleasure to introduce Janelle to you. Janelle Talavera is an undergrad here at CSUN. She took this class in spring 2017, so just the previous semester to this one. And during the middle of the semester, she came to me and she said, I really want to do research in evolution. I love evolution. How can I get involved? What can I do? I'll do anything and everything. And she really did. She took every single opportunity that came her way and has just grabbed these opportunities with such passion and enthusiasm that we wanted her to come here and tell you a little bit about the research that she's doing with Dr. Gray, who's in the back. You might know him. He teaches evolution, field ecology, the desert biology class, which is a class I want to take one day. So that's Dr. Gray. She's also working with Emma, who you know, because she is your PLF for this class. And so Janelle's going to give you a short presentation on the research that she's doing as a way to illustrate the kinds of research that you could get involved in and to sort of encourage you that this is a good time now. You're taking 106, you can start doing research. You find someone who can support your interests and we'll go from there. So let me attach this to Janelle. You put this on somewhere in your belt. And then this.
All right, cool. All right, thank you, Dr. Robertson, for that wonderful introduction. So hello, everyone. My name is Janelle, and today I'll be talking about the research I'm doing, which is the call analyses of Gryllus saxatilis and Gryllus navajo. So just to give you guys some background information, here are our two crickets under study and each of their habitats. On the right, we have Gryllus navajo, and their habitat is mostly red clay soils, whereas on the left, we have Gryllus saxatilis, and their habitat mostly consists of dry, rocky slopes. And as you can see, there's quite a contrast and difference in coloration between these two habitats. And what's really cool is that each cricket matches with their habitat pretty well. So Saxatilis and Navajo range from throughout all of Western USA. And so here I have a map, and the black circles represent Saxatilis, and the red triangles represent Navajo. And so Saxatilis occurs throughout California, the Mojave Deserts, the Great Basins, whereas Navajo are restricted to the painted desert regions of southeastern Utah and northeastern Arizona. So here's where it gets really interesting. Where Saxatilis occurs, they're uniformly black, but it's Navajo. These are the crickets that exhibit a color polymorphism, which basically just means that there are some Navajo crickets that are black and there are some that are red. So they range in colors from black to red. And so the question is why? Like, why do we have these black color morphs? And what's really interesting that I forgot to mention is that Saxatilis have wings, so they're able to fly into Navajo territory. And so is there gene flow happening? Is there hybridization occurring? And so to test that, Emma is doing a genetic analysis from multiple populations from both of these cricket species. And I, another undergrad, Sean, and Dr. Gray, are analyzing calls from also multiple populations from both of these cricket species. So why study song? I'm sure you guys have already learned about sexual selection and sexual advertisements and reproductive isolating mechanisms. And so cricket songs have also been well studied in this regard. So you all know that animal mating signals, they're typically species specific. And so those could serve as very important barriers to gene flow to keep two species ecologically divergent or separate, different. And so in studying these calls, if the calls are different, then yes, they can serve as barriers to gene flow and hybridization would not be occurring. If the songs are the same, then no, they are not serving as barriers to gene flow, and hybridization could be occurring, and it could be a possible explanation for why there are these black color morphs of Navajo crickets. So here I have two songs, one from Saxatilis and one from Navajo, and next to it I have an image of the song. And what's really cool about doing this research is that, yes, we could hear the song, but it's to be able to actually see the song and see sound and measure sound, that's really cool, I think. So I'm just gonna play Saxatilis for you really quick first. and then I'm going to play Navajo. And so you guys are probably all like, what the heck? They sound the same, right? Well, maybe there's differences in the song that female crickets can pick up on, but we can't as humans. And actually, if you take a look at the photos right now, there are a few differences. As you can see here, Saxatilis, those ovals you see are pulses, and this one has 3 pulses per chirp, while Navajo has 4 pulses per chirp. So already there is quite a difference in song. So in analyzing these calls, we've analyzed 112 Navajo songs and 80 Saxatilis songs. And what we do when we analyze these songs is that we measure different traits of the song. Traits such as interchirp interval, pulse duration, interpulse interval, pulse period, chirp duration, and chirp period. And so once the data is collected, the data will be able to tell us whether these songs are different enough to serve as barriers to gene flow. So we have 2 parts to our results. Part 1, some traits were not different. These traits included chirp rate, pulse per chirp, interchirp, pulse duration, and frequency. And part 2 of our results shows that two song traits were different. And so this is where it gets a bit tricky. Statistically, yes, they're different, but they're not significant in a biological sense, which basically just means that, I think the best way to just explain this to you is to give an example. So let's say gene flow was happening and Saxatilis males were flying into Navajo territory. Would those females be able to differentiate the Saxatilis songs from their own species male songs? Well, based on my data and based on my results, no, because it's just not different enough. So just to tie it all in, even though yes, there are some differences in song, they are not serving as barriers to gene flow, and therefore hybridization could be occurring, and could be a possible explanation of why there are these black color morphs. We will know for sure when the genetic analysis is done by Emma, and then it will let us truly know if those black color morphs are hybrids of Saxatilis and Navajo. And so you guys are probably wondering, why are you guys studying crickets and why are you guys studying evolution? Well, we learn about evolution in a classroom, but if you really think about it, it goes beyond that. There are many fields that have implemented and integrated evolution into their own studies and research. Fields such as agriculture, forensics, psychology, sociology, and medicine. I mean, there's even a whole branch of medicine called evolutionary medicine. And it just comes to show that, yeah, evolution, it's weird and it's sometimes really complicated, but it's beautiful and it's vital. And for us as a human species to move forward, evolution must be studied. So thank you guys. I hope you enjoyed this presentation. If you have any questions, probably you won't have time, so she'll answer them or something. Okay, later. Yes?
So you're not a cricket?
No.
So how do you know those are biologically linguistic differences?
That's a great question. Let's go back to the slide. Well, it's just it's way too small of a measurement. So let's say I'm 5'10", and there's a person who's like 5'10" or 5'9.5". There's like barely a difference, right? So you can't really tell, but so that's what it kind of means.
But how do you know? Because you're not a cricket.
I don't know. I'm sorry, I don't know.
No, it's okay that you don't know. We'll be talking about this a lot, I'm sure. But how, and maybe the class can chime in as well, and think about how would you test whether these are biologically meaningful or not? How would you test that? You might have some. So then, put it around predators. Let's see if the predators
Okay, well, we could just, oh, can I answer now? Okay, so we would have Saxatilis and Navajo, and basically see if the female would respond to a Saxatilis call rather than its own species, like put them together in that sort of way. Sorry, I'm not explaining it very well, but along those lines.
What do the rest of you think about that? Do you think that would work as a way to test whether it's biologically meaningful? Basically, make choice study. Like, it's not that different from all the studies that we looked at that were, do males and females prefer males with long tails or short tails, right? Sarah has a question.
How do we prove to be for sure that there is no interference?
We don't know that yet. That's still under study. Right? Okay. So Emma is the one, she's doing a genetic analysis to make sure that's happening. If not, so.
Any other comments or questions?
Yeah.
Have you studied any other organisms, or is this like your first?
This is my very first.
Do you love it?
I love it. I am in love with evolution. I love all the research I'm doing. And so I'm really glad I changed my major to evolution. Yeah, actually this last summer I went out in the field with Emma. We spent a week at a time. It's great, scaling rocks, falling off rocks, it's great. So we used a program called Audacity, and so this is what the program Audacity would show the song as, and then we would just measure like each, we'd count the pulses per chirp and then measure how long each pulse would be, and we just log in the data into like Excel and to keep track of our data. I'm sorry? The way that Dr. Gray records the songs, he has a special apparatus so that background noise would be canceled out and you can just focus in on cricket song. Yep. I didn't pick crickets, crickets found me. That's really what happened.